Hi, and welcome to today's seminar. Today, Dr. Neil Singla will present Bunionectomy Part 1, the model overview, both pros and cons. Dr. Neil Singla is the founder and chief scientific officer for Lotus Clinical Research, established in 2001 as an analgesic site in CRO. The company has 40 employees, a 40-bed Phase 1 unit, and the ability to run Phase 2 and 3 analgesic programs on a site and/or CRO level, full CRO capabilities, and most importantly, therapeutic expertise in pain. Dr. Singla currently chairs the Analgesic Clinical Trials Special Interest Group at APS and IASP. The main focus of Dr. Singla's academic endeavors has been to analyze and understand how the inherent variability in analgesic clinical trials can be minimized. Dr. Singla consults for multiple pharmaceutical companies regarding U.S.-based regulatory analgesic issues. And now, without further ado, here is Dr. Neil Singla.
Thank you for that introduction. I'm excited to get started today. The topic at hand is bunionectomy as an acute surgical pain model. As I started to put the webinar together, I realized that the topic of bunionectomy is relatively large and is probably more appropriately covered in two separate webinars. The first is the webinar that I'm going to give today, which is an overview of the model in general, talking about where the model is strong and where the model is weak. This webinar is really geared toward people who are in the throes of making the decision of what acute surgical pain model best meets the needs of their drug. The next webinar, Bunionectomy Advanced Principles, Part 2, will discuss the model in detail when the choice has been made about surgical model and that choice has centered on bunionectomy. Then there are detailed methodological questions that one begins to ask themselves. And those questions will be the focus of the Part 2 webinar. Questions like, what is the pain trajectory of bunionectomy on post-op day 0 dosing versus post-op day 1 dosing? What can you do to mitigate placebo response? What is the appropriate use of rescue medication in bunionectomy? Which drug should you choose? And how can you investigate opioid-sparing properties of a drug in this model? The basic problem that one faces when they're trying to experiment on a drug meant to control acute post-surgical pain is that acute surgical pain requires that a patient have surgery. That's obvious. And I classify that obvious fact here as a problem because the surgical procedure itself and the clinical requirements in order to achieve a safe and positive surgical outcome confound the analgesic methodological considerations of the experiment. In other words, as physicians, our first responsibility is that the subject have an excellent and safe surgical outcome. But in order to ensure this, we have to make compromises that make the experiment less clean. Our hospital here in Pasadena has 17 operating rooms, and when I walk in and I see the board full of cases, I think to myself that there are a huge number of choices that I have in order to experiment on surgical pain drugs. Almost all of those patients on the board are going to have post-surgical pain. But in reality, if your goal is to run an experiment that has been run before and therefore is understood, you really only have four main choices. If you choose a model that's not well vetted, then your experiment may have error that you did not account for, and this may lead to a false negative outcome. So after considering various models, most people revert back to one of the four well-understood models with the general idea that it's better to deal with the devil you know than the devil you don't. And if you decide that that mindset is correct, then you really have four main choices, which are dental extraction, bunionectomy, joint replacement, either knees or hips, and soft tissue surgery as the fourth. These models all have their strengths and weaknesses, but if you choose one of these four well-vetted models, you will at least be able to look back at past studies that have been done in these models to understand the key methodological principles. Now, the next concept is to divide these models into the method in which patients are accrued into them. We're going to call one method the recruited type models and the other non-recruited models. This webinar being a scientific discussion may make it seem odd that we would talk about methods of recruitment. But in fact, the manner in which subjects are accrued for each of these models has a significant impact on the scientific and methodological considerations. That being said, let's talk about the terminology. What is a recruited model? In recruited models like dental extraction and bunionectomy, the surgical and anesthetic fees associated with the procedure are subsidized by the research grant. What that means is that subjects are accrued through advertisement, newspaper advertisement, radio advertisement, Facebook, social media of other types. And because the fees are subsidized, there's increased freedom in regards to health networks, meaning that it really doesn't matter what type of insurance a patient has, they can still come to Research Center X and have the procedure. And of course, this is a very nice perk for subjects who may have high deductibles and may want to have their surgery done in a timely fashion at a specialized research center. And so essentially what all of these factors end up doing is they allow funneling of subjects into specific research units. I'm going to continue with this topic and pull out a few critical data points. If one is interested in the source of this data and or the method in which it was collected, please refer to our comprehensive review published in IASP's journal last year. This graph shows a relatively simple metric, which is enrollment rate. What we did is we just looked back at all the dental studies, bunion studies, joint replacement studies, and soft tissue studies that actually published their enrollment rates. A lot, but not all articles will tell you the time it took to recruit the number of subjects that were recruited and how many centers were used. And if we had all of that data, then we calculated a subject per center per month number for each model. And what we found is displayed here. One can see that dental studies generally will enroll 63 subjects per center per month, which is just a very large number. Bunion studies, 20 subjects per center per month, joint replacement 3.9, and soft tissue 2.4. These are really large disparities in enrollment rates. Anyone who's been in the pharmaceutical industry for a while knows that projecting site enrollment can be tricky. So having an objective feel of what will most likely happen in a certain model is very helpful. Let's examine this hypothetical study, late phase 2 or phase 3 acute pain study. Typically will have a N of approximately 240 patients, and in general, a nice recruitment time is about 6 months. Under those parameters, if you're doing a joint replacement study, the math necessitates that you need about 10 centers, whereas if you're doing a bunionectomy study, you only need 2 centers. And this fact is relevant not for business purposes, but for methodological purposes that have to do with variability reduction. And before I go too much further, I'm going to provide what I call a disclaimer in reverse, which is that, of course, I have my own proprietary interest in Lotus Clinical Research as a site and as a CRO, but Lotus performs all 4 models. So the purpose of this webinar is not to sell or promote bunionectomy. I want to fairly evaluate it, where it's good and where it's bad. So we're going to get to both components of that. So the first point is that if you have fewer centers, then you have less variability. There's fewer surgeons, anesthesiologists, recovery room personnel, study coordinators, which leads to a cleaner experiment. In addition, the convalescence after bunionectomy is not as complicated as the convalescence after joint replacement. Patients who undergo joint replacement need pretty intensive physical therapy. They need a hospital environment with a lot of different types of practitioners looking in on them. And all these practitioners add variability. They're giving drugs. They're taking the patients down to MRI. They're doing X-rays. They're doing exercise therapies. They're putting patients on continuous passive range of motion devices. All of this stuff affects the analgesic outcomes that we can measure. In addition, the demographic of bunionectomy patients compared to the demographic of joint replacements is that they're younger and they have far fewer acute medical needs. In addition, the setting that the research occurs in for bunionectomy versus joint replacement is completely different. Because of the acute medical needs of a joint replacement patient, those subjects need to be in a hospital environment, whereas bunionectomy patients can have their operation at a surgery center, but then be transferred to a highly specialized research unit. In acute surgical pain research of any sort, there are two masters. One is clinical care of the patient, and the other is the study protocol. In a hospital environment, it's very clear which of these priorities will win out, and that is local care. In a research center, it's absolutely imperative that the subject receive excellent medical care. But the manner in which that care is achieved is much more flexible than one would find in a hospital environment. In the same methodological review that I pointed to earlier, we had another very important data point, which was the signal-to-noise ratio generally found within the different models. On the y-axis, we have the standardized effect size, which is a common metric used to look at the signal-to-noise ratio or the cleanliness of an experiment. And on the x-axis, we have our four very common models: dental, bunion, joint replacement, and soft tissue. Again, we simply pulled papers, looked at the primary endpoint of interest, calculated the standardized effect size, averaged them for each model, and put them into this graph. What we found was quite startling, that all of the theoretical discussions that I've just had regarding recruited models and why they are potentially cleaner from a scientific perspective are in fact very clearly borne out in the literature. The standardized effect size of the recruited models is far greater than the non-recruited joint replacement and soft tissue models. This next slide is essentially, the data from the previous slide with one new column, which is requisite N. If one wishes to have 80% power to obtain a p-value of 0.5, and then you know the predicted effect size of your experiment, you can calculate the requisite N. The N is inversely proportional to the square of the standardized effect size, and therefore small changes in the standardized effect size can have major differences, as we see here with third molar extraction versus visceral surgery or soft tissue surgery. A change in the effect size from 1.4 to 0.4 changes the number of patients that you need in the study from 30 to 200. It's probably important to quickly discuss some of the common misperceptions about the bunionectomy model. The most common one that I hear is that bunionectomy is an outpatient surgery and it's really not that painful, which in general is actually true. But when the model is performed under the correct experimental conditions, the surgery does have the requisite pain to evaluate not only weaker oral drugs, but also very strong intravenous analgesics. Also, the model has a vetted history of regulatory acceptance. To hazard a guess, I would say that 90% of the drugs that have been approved for acute pain since the year 2000 have used bunionectomy somewhere in their registration process. And the model itself has a lot of pain because the surgical procedure requires 2 osteotomies. And the periosteum of bone is very highly innervated, and that's why it's so painful. And it's also why the model tests NSAIDs really well, because NSAIDs work well on bone pain. Okay, so now that we have talked extensively about where bunionectomy is really strong, let's turn our attention to where it's weak. If you compare bunionectomy to dental extraction, you'll see that the standardized effect size in dental extraction is actually much higher. So not only is the signal-to-noise ratio in dental higher, which means that you need less patients in a dental study, but also those patients are significantly cheaper. The dental surgery itself just doesn't cost as much as bunionectomy. You don't need as sophisticated of a surgery center, and you don't need an anesthesiologist. Because of these factors, the recruitment speed is much quicker for dental. So for all those reasons, dental is a great proof of concept model. It's cheaper, it's faster, and it's more sensitive than bunion. However, if you're looking for a multi-day experiment, bunionectomy has good assay sensitivity from 0 to 48 and up to 72 hours, whereas dental probably has good assay sensitivity for only the first 24 hours. A large portion of the previous discussion was spent explaining why joint replacement was, from an experimental standpoint, inferior to bunionectomy. But there are situations in which a joint replacement study is superior to bunionectomy, and let's go over those. First of all, if the real-world utilization of the IP is going to be in a hospital environment, then you get more information from a joint replacement study, which is actually performed in a hospital, than you would in a bunionectomy study. If one expects that when the IP is actually used, that it will be used in subjects that are greater than 60 years old and match the demographic of a joint replacement experiment, then of course you're going to get better clinical information from this type of experiment than you would from a bunionectomy study. And what this ultimately leads to is better acceptance of the study results from the end user of the product, which is the clinician. Convincing orthopedic surgeons to use a certain drug based on bunionectomy data is much more difficult than it would be if you had the same data in subjects that they are doing surgery on in the hospital environment, which is knee replacements and hip replacements. Wow, after going through this, I really am glad I broke it into 2 webinars. I hope that there were some people at least that were able to stick with me through the whole thing and that found the information useful. Bunionectomy Part 2 will be coming up soon, hopefully, and I thank everyone for listening.